Good morning. Today we are talking about our first energy pathway called cellular respiration. There are 3 energy pathways we will talk about at the end of this cellular unit: cellular respiration, fermentation, and then photosynthesis. So today's topic is cellular respiration. This is the aerobic process of breaking down sugars and foods that we eat to gain something called ATP, that form of energy that is the spendable kind of cash of the cell. ATP stands for adenosine triphosphate. We talked about this in the energy lecture, so look back at that if you want to look at the chemistry of ATP. So really, cellular respiration is about taking the foods that we are eating and breaking the bonds in those foods and creating this ATP, this form of energy that we can use to do our life work. The overall reaction for cellular respiration, if we use a sugar, is a sugar like glucose plus oxygen is going to create CO2, a waste product that we exhale off, excess water that we also can urinate and sweat off, and then this ATP, this sort of energy form for the cell. If you are going to do all 4 steps of cellular respiration, you need a mitochondria. And a mitochondria, it includes, I'm going to mention a few different key pieces, but this internal space with the liquid is called the matrix. And then this inner membrane folds and creates a little space between the inner and outer membrane. So those are all important structures for cellular respiration. All right, you might have heard of respiration before. Respiration has to do with your lungs, and I want to just kind of come back to how it's connected to cellular respiration. Sometimes students confuse these things. So respiration is the breathing or ventilation of the lungs, allowing us to take in oxygen and release or expel CO2. Cellular respiration is an energy reaction that happens in our cells. And this energy reaction is going to happen so that we can break the bonds in food and create ATP, the energy that we need to do our life work. However, respiration is actually required for this reaction. So the reason you breathe and do respiration is to do cellular respiration. This reaction, this cellular respiration reaction, requires oxygen. So we need to breathe in oxygen and bring it to our cells so they can get energy. And then one of the waste products of this reaction is CO2, or carbon dioxide, that we're going to breathe off. So the reason you breathe is cellular respiration, but the 2 processes are connected. And cellular respiration is the energy reaction that happens in all of your cells. So keep that in mind as we go forward. I want you to know the 4 steps of cellular respiration and their order. So step number 1 is called glycolysis. We're going to call step number 2 pyruvate oxidation. It has a few different names, but that's what we're going to call it. Step number 3 is citric acid cycle, and step number 4 is oxidative phosphorylation. So make sure you know those steps. I also want you to know where in the cell these steps are occurring. So glycolysis, step number 1, happens in the cytosol, or the liquid part of the cell. It actually does not require the mitochondria. So it is probably a very evolutionarily old step because it's something that both prokaryotes and eukaryotes can tweak and use, and all you need is the liquid part of the cell. So we'll be talking about that a little bit. It's also the first step of fermentation, the second process as well. So glycolysis happens in the cytosol, or the cytoplasm of the cell. Pyruvate oxidation, step number 2, happens as the sugar crosses into the mitochondria. It comes through a gate or a doorway. And as it goes through that doorway, the chemical is actually altered. And so pyruvate oxidation, we say, happens as the sugar crosses into the mitochondrial, or across the mitochondrial membrane. The third step, citric acid cycle, happens in the mitochondrial matrix, which is the liquid center portion of the mitochondria. And step number 4, oxidative phosphorylation, occurs in that inner membrane space, or on that inner membrane of the mitochondria. So these are the 4 steps. Make sure to know the order of them and where they occur in the cell. This is also on your study sheet, so you can follow along and fill this out as you're going through this lecture. All right, I want to take you through the 4 steps. I want you to know both what you're starting with and what you're ending with. I want you to know the location of where this reaction happens, and then how much ATP is made, because the whole point of doing this process is to make this energy form, this ATP, so that we can do our life work. So we're going to track ATP in this process as well. So for glycolysis, it's actually naming what it's doing. glyco for glucose, and lysis for splitting. I'm going to take you through this reaction starting with glucose. And so we start with glucose, and I have 6C in parentheses on this note page to remind you that glucose has 6 carbons to it at the beginning. We split this sugar in glycolysis, and when we split this sugar, we start with 6 carbons, we split it into 2 3-carbon pieces called pyruvate. And when we split this particular sugar, we gain some ATP when we do this. And that ATP is that energy that we can use for our life work. And we also make something called an electron carrier that will come up in our very last step number 4 of this particular reaction. So we start with the 6-carbon glucose, we make 2 3-carbon pyruvate, a little bit of ATP, and an electron carrier. Again, this does not require an organelle. It happens in the liquid portion of the cell. I want to sort of direct your eyes over to this flow chart over here. I want you to notice that this particular first step, glucose is a little bit of an uneven molecule, and one of the first things that we have to do is we have to even it up so that the 3 carbons are equally split or easily split. And so we actually have to invest 2 ATP at the beginning to rearrange glucose in order to split it evenly. When we split glucose, then we get 4 ATP, but we often write this as a net ATP, 2 ATP gain, because we're applying 2 to start the reaction, we're gaining 4, so we're actually making a net of 2 ATP by the end of this particular reaction. So keep that in mind. It's kind of that old adage that you have to like spend a little money to make a little bit more money. So something like that is happening in and around your cells. So glycolysis, we split our glucose into those 2 pyruvate, and we make a couple of ATP along the way. As the glucose or the pyruvate and our sugar is entering the mitochondria, these 2 pyruvate will go through a gate. And as it goes through a gate, number 1 is showing that as it comes through this gate, one of the carbons, remember it has 3 carbons left, is going to be lost as CO2. In our original glucose, we have 6 carbons, and we're going to lose all of those carbons as carbon dioxide, a waste product that we exhale off. And so we split off one of the carbons for each of the pyruvate. We make a little bit of an electron carrier, 1 electron carrier, and then onto this 2-carbon piece, we are going to put a vitamin. It's actually a part of the vitamin B complex. Sometimes vitamin Bs are sold as energy vitamins. And so the reason why is because that vitamin B, one of them, is going to move on to the top of these 2 carbons to be an acetyl-CoA, the CoA or coenzyme A group. And so this then builds this acetyl-CoA. Acetyl-CoA is the baton that gets to move to the 3rd reaction. So again, we started with our 3-carbon pieces called pyruvates. We send them each through this reaction, so we're actually doing this twice. So we're making a total of 2 acetyl-CoAs, so 2 of them, 1 for each of the pyruvates. We're making a couple of CO2s and a couple of electron carriers. This reaction happens, we say, as the pyruvate or the sugar is crossing into the mitochondria. Step number 3 is called the citric acid cycle. This acetyl-CoA group up here, you can see it has the 2 carbons left with that coenzyme A attached to it. And it actually comes along and bonds with a 4-carbon piece called oxoalloacetate. These bind together, this receiver molecule binds the acetyl-CoA and becomes citric acid. So that's why this cycle is named the way it is. It's like the starting chemical for this particular cycle. Acetyl-CoAs, then we cut off or break off the carbon groups on that acetyl-CoA group, so we lose the rest of our carbon dioxide. We're going to run this particular system twice, and so we're going to make a total of 4 carbon dioxide, 2 ATP, 6 NADH, and another electron carrier that goes to the last step called FADH2. And so these are what we end with. The batons that get to move forward to the last step are these electron carriers, the NADHs that we've made in every single step, and the FADH2s. So this is step number 3, the citric acid cycle, and it happens in the mitochondrial matrix, or the liquid part of the mitochondria. Step number 4 is composed of 2 different steps, but I'm always on a test or a quiz going to call it one thing, or oxidative phosphorylation. So I'm going to put both of these steps underneath that particular title or name. The 2 steps, so all of the electron carriers that we have made, we've made NADHs in step 1, 2, and 3, and then we've made some FADH2 in step number 3. All of those NADHs and FADH2s come to this last step, and they deliver their electrons, or these H's. H's they have a single proton and a single electron to them, right? A hydrogen, if you think about hydrogen and its atom, they deliver those sort of electrons to the set of proteins that sit on the inner membrane of the mitochondria. The first thing that's going to happen is this H gets pulled off, and the H's will be pumped across into the inner membrane space. This is a proton pump that we are setting up. The electrons are separated, and as the electrons are passed down this chain, they give up a little bit of energy, and that energy enables this pump to pump these H's against their gradient. The last sort of electron acceptor in this reaction that pulls the electrons that have been used away from these proteins is oxygen. Note that this is the only place that we use oxygen in all 4 steps as this electron sort of acceptor at the end of the electron transport chain. Now, step number 2, once we have all these hydrogen outside in this inner membrane space, then we can send them through an enzyme called ATP synthase. And ATP synthase is like a little turbine on the inner sort of membrane space, and it actually spins. And as it spins, it's able to put ADP with some phosphate groups that are loose inside of the mitochondria back in and reform ATP. This particular step makes a total of 28 ATP. We say that cellular respiration is aerobic, requires oxygen, because so many ATP are made in this last step. If you remember, only 2 ATP are made in step number 1, 0 in step number 2, another 2 are made in step number 3, and then 28 are made in this last step. Because this last step requires oxygen but makes most of the ATP, we say that the whole process is aerobic, although in details that's not really true. Just this last step would require oxygen. Now, when we look at what I've shown you, I have shown you how to complete this particular reaction. I want you to know the total ATP made in each step. I want you to know kind of the starting and ending reactions and that energy that is made. So make sure you know those things, and make sure you know the total. If we add the 2 plus 2 plus 28, we get about 32 ATP made per every glucose, right, that we send through this particular system. If we're using glucose, you also start with 6 carbons in your sugar, and you're making 6 CO2 because you're losing each of those carbons as like sort of carbon dioxide. But I also want you to note, I've shown you how to do this with sugar, but I want you to note that you can actually run this reaction with other foods like fats and proteins and other kinds of sugars. You have to tweak kind of where you're entering and the enzymes that you're using, but overall you can actually do this reaction with other foods that you make as well. Now on your study sheet, you actually have a table that I want you to summarize like the steps. You'll have a table for cellular respiration, one for fermentation and one for photosynthesis, but I also have that here so that you can kind of take a look at how this reaction goes down and what's being made in each reaction at a single glance. So I will see you next time when we discuss fermentation.